Yes, very happy to join and share our work on SalivaDirect. So this all started back in March when an amazing group of sort of researchers, clinicians, scientists from all over the Yale School of Public Health, Medicine, Yale New Haven Hospital got together to form the Impact Biorepository. And so this was the idea that as we could see the pandemic approaching, but we we're going to try and enroll COVID-19 inpatients into our biorepository to build up a bank of samples so that we could better understand SARS-CoV-2 and COVID-19 disease progression. So this helped us lead to SalivaDirect. So what happened is that during this, as we saw the outbreak continuing, we saw that saliva was working very well for SARS-CoV-2 detection, and we were very aware of needing to have better access to testing and cheaper testing opportunities. So you may have heard of SalivaDirect. We did receive FDA emergency use authorization on August 15th, and when it was issued to us, it was described as the most unique EUA that has been issued to date. This is because SalivaDirect, first of all, is not a test kit. We're not providing anything, we're not packaging anything together, we're not making anything, we're not distributing anything other than the protocol for the test. So what the FDA has enabled us to do is to take our validated test and be able to designate other labs under our authorization to be able to use SalivaDirect to test saliva samples for SARS-CoV-2. What's also really unique about our EUA is that as we update the method for SalivaDirect, this is going to extend to all other labs. So again, SalivaDirect really arose from wanting to reduce testing costs, increase the number of tests in the community, and reduce the implementation time to use saliva. So I think all of us are familiar with the gold standard nasopharyngeal swab. This really came about very early on in the pandemic. We sort of had to default, we had to default to what we knew very early on, and the nasopharyngeal swab was already used for so many other viral and pathogen panels in clinical diagnostics. So, we just had to hope that this was going to be ideal also for SARS-CoV-2. And indeed, it did work well. In fact, so well that actually sometimes a paired oropharyngeal swab would be taken, but this was quickly removed because of supply chain issues with the swabs. So really defaulting on that nasopharyngeal swab. And it does, however, require a trained healthcare worker to take this. It is, as you see, it is considered an invasive method. Therefore, it is uncomfortable to the participants as well. And due to the how uncomfortable it is, it could cause the patient or study participant to sneeze or cough on the healthcare worker who is taking the sample, which puts these healthcare workers at risk. These samples are then traditionally transported to the lab, as they were for us, and then they're processed by RNA extraction. So you take the viral transport media, you perform RNA extraction to release that genetic material of the virus so that you can try to detect it. This, however, is a very expensive step. It's labor intensive. And as what we saw for the nasopharyngeal swabs very early on, the RNA extraction kits were actually falling into supply chain issues as well. So we're seeing shortages for both the nasopharyngeal swabs and the RNA extraction kits. So once we have that RNA, we were then testing them in RT-PCR for two SARS-CoV-2 genes, N1 and N2. These were described by the CDC. However, we were ordering our primers and probes from other sources because, like the RNA extraction, we wanted to make sure that we were not using things that were really important to the clinical supply chain. So we wanted to make sure that we're drawing from other supply chains so that we wouldn't have shortages. Well, we wouldn't add to the shortages that we were seeing in the hospital. Testing for those two SARS-CoV-2 regions, N1 and N2. We were also testing for human RNase P. That's tested for as a quality control measure. But all of these were having to be run individually. So this was also a very labor-intensive and expensive step. And we actually found that N2 was a lot more noisier than N1, that this would often lead to, wouldn't often, but if we were ever to have an inconclusive result, this would be coming back down to N2 as compared to the N1. So it was really, it was the supply chain issues really that drove us to look at an alternative sample type. But we were also very aware of just how expensive and time consuming this whole process was as well. So as we started to see the results coming back from the Yale biorepository, so what was happening is that we were receiving a whole range of samples from COVID-19 in patients. They were just to be banked in this biorepository, but every sample type, whether it was the nasopharyngeal swab, saliva, urine, rectal swabs, we had throat swabs for a while as well, they were being tested for SARS-CoV-2 present in this RNA extraction and RT-PCR detection. And we started to see that saliva was performing pretty well. And actually, this is a slightly outdated slide. This graph now appears in our New England Journal of Medicine paper. And what we found is that these are samples from COVID-19 in patients, and we did actually see a higher viral load detection in saliva as compared to the nasopharyngeal swab. We did see this sort of happening quite early on, but we didn't want to sort of get ahead of ourselves. We really just thought, saliva is not really a traditional diagnostic fluid, but we decided to keep an eye on it because just knowing about all of those supply chain issues that we were facing with the swabs, we knew the hesitancy of the healthcare workers especially with taking secondary swabs for research. They'd already put themselves at risk for the diagnostic swab, but then to ask them to go in again, they weren't always the happiest about that, and very understandably, especially when they were rushed off their feet. So the idea that, saliva was looking promising and that it could get around a number of these issues, that's really what we decided to study forward, particularly if we could use it to, we knew that we were going to be facing more frequent repeat testing needs, and that we knew that people were not going to be wanting to have that nasopharyngeal swab on a, every 2 days, every 3 days. So that's why we really decided to see what we could do with saliva. So we came up with SalivaDirect. So as the name suggests, again, we are still taking that saliva sample, and then we're testing it almost directly in RT-PCR. What we do is this saliva sample is mixed with an enzyme called Proteinase K, and we just mix that. If you're doing it individually, you can do it for 30 seconds. We do, because we vortex a whole lot at the same time, we do it for a minute just to make sure that Proteinase K can really get in there. It can break down the proteins of the saliva. It can maybe start breaking down some of the virus. And then we just heat and activate that. We heat and activate that for 95 degrees at 5 minutes. That's to inactivate the Proteinase K before we put it into RT-PCR detection. We've dualplexed the RT-PCR, and now we are only targeting the N1 and RP. Again, the N2 just gave us a lot of noise, and N1 is being used all over the country at the moment, and it's constantly being followed for any potential changes that could maybe affect its sensitivity. We just thought that for us at the moment that this was working really, really well in our hands, that we felt sort of confident just moving ahead with N1. We do still detect RP, so we keep RP in there again as the sample quality control. So this is detecting the human RNase P. And this is just to tell you that, say for example, if your N1 is negative, we need to know that the RP is positive to make sure that the saliva sample was in good shape, that everything went through the process properly, so that if your RP was negative, you'd know that something was wrong, and that would be the reason potentially for your negative detection of N1. So some very key points that we looked at into this process is that, first of all, we found that saliva is stable without preservatives. This was really important for us for keeping the prices down and to increase accessibility to testing. A lot of the saliva, there's not a lot of them, but the few saliva tests that are out there, many of them rely on these very expensive, fancy collection devices, which are full of stabilizing buffers, which are they're marketed to stable the SARS-CoV-2 RNA. But these tubes can range from anywhere from about $7.50 to almost like $30 per tube. Whereas if we just use very simple laboratory plastic tubes, these are sort of 10, 20 cents. So you can imagine if you're setting up a mass testing strategy, you're going to price out much earlier if you've got $7.50, $10.15 tubes versus 10 or 20 cent tubes for sample collection. So what we did for this is we did, just to confirm this, we took a highly positive saliva sample and we diluted it down to 12, 25, and 50 virus RNA copies per microliter. And then we just incubated them for 7 days at 4 degrees, room temperature, and 30 degrees. And so this is the same sample in those different settings. And you can see just very, very constant detection of SARS-CoV-2 N1 in the raw saliva sample. So this really helped encourage us that no expensive collection device was required. As a little bit of an aside, we did actually expand this out further, and basically each of these dots joined by a different line, by the dotted line, are the same sample. And so again, these are using clinical samples, and again, we saw stable detection in room temperature for 5 days, 30 degrees. But when we looked at the graph on the right, these are samples that were kept at room temperature. And again, going out, there's one down the bottom that you can see, going out all the way to about day 25, SARS-CoV-2 detection in saliva was remaining stable. So again, just really supporting that we don't need these expensive tubes. It was really important to us to keep SalivaDirect as sensitive as possible. I know there's a lot of discussion now regarding about the sensitivity, and especially of PCR, and the relevance of this. But we started developing SalivaDirect very early on in this all, when we didn't actually really know what the cause for concern for low viral loads may or may not be. And at that time, we just really wanted to make sure that our assay was going to be as sensitive as possible, and that we would try and catch as many infections as possible. So that's how we really just developed SalivaDirect, is that we were comparing the CT values, so the virus detection that we would get when we sent a sample through the full RNA extraction process versus the simplified SalivaDirect process. And we did actually see very consistent detection between these two processes. We also learned quite early on, which you can see there's a sample here that's around 30 CT and drops down to being negative in SalivaDirect, that unfortunately SalivaDirect is sensitive. So far, the only thing that we've seen it's sensitive to in terms of inhibitory substances are blood and mucus. So those were, unfortunately, those are the things that SalivaDirect is sensitive to, but so far looking at different foods and things, we're not seeing that same result. But that was just good for us to be aware of and thinking about in what sort of populations you want to be using SalivaDirect as compared to the RNA extraction. So again, this just really helped support us that we could remove this expensive and time-consuming step. Again, we just also wanted to make sure that by going into the multiplex PCR, that again, the assay was going to remain sensitive. So this was sort of our early validation work when we were using a multiplex as compared to the dualplex. So when we had the N2 in there, again, this has remained stable with us taking the N2 out now. So we are able to achieve low limits of detection, and we have that internal control of RP in there. I mean, as I said, N1 is being used by so many labs all around the country, and it's looking highly specific, but we did just have some saliva from prior to the COVID-19 outbreak. So we had saliva samples collected from the autumn-winter season earlier this year before COVID-19 hit us, and also the year before. So we just wanted, again, just make sure that we just ran these samples through just as a double check to make sure that we weren't going to be detecting any N1, like there wasn't going to be any cross-reactivity with other upper respiratory tract bacteria or viruses or anything. So one of the things that I mentioned was very unique about SalivaDirect is that we're able to expand our method. We've already done this to start with, and that's by validating reagents and instruments from different suppliers. So for each of the steps, and the few that there are, adding the Proteinase K, the PCR kit you use, and then the instrument that you run your RT-PCR on, we've already validated them from different suppliers. The idea of doing this was, first of all, to be very mindful of those supply chain pressures. We didn't want either to sort of release our method and to see one supplier just get totally overwhelmed, or of course, if one of these things became key into someone else's, like one of the clinical diagnostic chains, we wanted there to be alternatives so that it would remove pressure there. We also don't want people to be having to really buy new things or order new things. So we're hoping that, we wanted to start with a little bit of a range of the PCR instrumentation already, hoping that labs would have these. And what we're going to do is we're already starting to continue and expand onto this. So we are able to validate more instruments using our method and add these to our EUA. Once we add them to the EUA, any of the labs who are designated under our authorization can then also use anything else that's on this list as well. So this will be constantly updated to help options, but this also helps to keep prices down, especially with that competition between suppliers. So at the moment, just the list prices of the reagents, it can range between about $1.30 to $4.30 in reagents per sample. But this is based on list price, that if you order in bulk, or if you've got good connections with the suppliers that we really think the cost should be able to get down just for the reagents for about $1 per sample. We are very aware that there are other costs associated with testing, of course, such as the lab space to run this in, the people to perform that, even the logistics of getting the samples. But we've managed this $1 per sample is at least 90% less than what some of the other RNA extraction based methods are. So we're hoping that that is a price that can really be passed on to those needing testing. We have maintained, as I said, a high limit of detection. So this was ranging in our EUA submission between, as you can see on the bottom here, around between 6 copies per microliter or 12 copies per microliter for our least sensitive combination. We did only stop at 6 previously. We have now carried this down to 3 and 1 copy per microliter, and we are sort of hitting around there as well. We've also recently received standards from the CDC, who have been sending these standards out to everyone who has EUAs to try and standardize what everyone is reporting. And actually, our method with SalivaDirect is the same as what the CDC's assay got as well for their limit of detection, which includes RNA extraction. So that just also gave us the confidence that we know that, yeah, that this is working well for us. We did have to do the clinical validation for this. So the first thing we needed to do is take paired nasopharyngeal and saliva samples and compare those first of all. So we did that in the ThermoFisher TaqPath COVID-19 kit. Again, we saw we had 94% sensitivity between the nasopharyngeal swabs and the saliva samples. We had some instances where the nasopharyngeal swab was negative but positive in the saliva, and then also an instance where it was negative in the saliva but positive in the swab. We then had to compare it with SalivaDirect, so we used those same saliva samples, and then we just tested those saliva samples instead via SalivaDirect and compared those to those nasopharyngeal swabs. And again, we saw a high agreement, 94%. And again, we had instances where, one of the sample types was positive and the other was negative. So, neither sample type is perfect, but also a lot of this discrepancy that you see in, this differences in agreement that you get, a lot of this is happening around the lower viral loads. So just, when individuals have low viral load and you're sampling them, if they've got very few copies for you to detect, maybe there are going to be more copies in one sample type than another. Maybe the same individual would test positive by one and then negative the next day anyway. So yeah, we had the 94% agreement, and this was satisfied the FDA's requirement to issue their emergency use authorization for us. One of the big questions, of course, though, has always been like, how do these tests perform in asymptomatic individuals? And this is actually where a partnership with the National Basketball Association really helped us to investigate this. The last few months have been absolutely mad, but I think an email from the NBA was one of those particularly mad and unusual days. And, while it did lead to some interesting NBA gear, it also allowed us to test we've tested over 3,800 saliva samples taken from NBA players, staff, teams. They're all coming de-identified, but they are matched to nasopharyngeal swabs. So it's a paired interior nares and oral swab, which has been used for their routine diagnostic testing. So we've been able to compare the performance of that routine test as compared to saliva with SalivaDirect. And we have seen high concordance with those nasal and oral combined swabs combined. And I think we can all agree that this asymptomatic testing, especially that the NBA is doing, has worked very, very well with the fact that they were able to pick up infections very early on and stop other outbreaks and be able to create the bubble that they have. So this information is also all available on our website. We've been very much living by the whole open science realm of this. We actually shared the first SalivaDirect protocol on protocols.io back in June. We've been sharing protocols before that. So everything is up there, also the validation results. And this is also where high complexity CLIA labs can come to us and request authorization to use SalivaDirect as well. So just a few slides in closing. Saliva does have potential as a sample type for SARS-CoV-2 detection. Again, this is also the collection of all oral fluids. So just anything so that sort of, if you think, maybe sometimes you have a bit of a nasal drip, mucociliary clearance, sort of everything that's sort of coming into the mouth as well as coming through the salivary glands. We're sort of just picking all of that up. While, there has been some hesitation behind this, again, it's not a traditional sample type, but other studies have shown a 90% concordance with nasopharyngeal swabs in detecting other respiratory tract pathogens, including coronaviruses, before all of this. What's really important about saliva is that healthcare workers are not required to collect saliva specimens, or that we can at least try to be moving away from that. So it really decreases the risk to those, but can also eliminate the waiting time for saliva specimen collection. If you can just hand out a bunch of cups to people, or you're in a classroom doing some screening, and every kid can just have their own tube, produce a saliva sample, rather than having someone to take all of those nasal swabs from them. So it is practical for efficiently screening a large number of individuals, and particularly when you're looking at frequent repeat testing, it is less discomfort to patients or just people who are being screened, and of course less risk to anyone who needs to be taking those swabs otherwise. So as we're seeing, outbreaks are going to continue, and they will disproportionately impact vulnerable societies. So we do need inexpensive, simple and scalable testing options. We do need to see a shift in this thinking about traditional diagnostic testing to tests that are more suited for screening and ongoing surveillance. So, these may be different to what we've seen in the clinical setting before, but if they are going to be more affordable so that more people can get tested, or they're going to be more suited to frequent repeat testing, those are sort of the tests that we need to be exploring more. And the benefits that I've also mentioned about saliva collection could also prove even greater in low resource settings, with the idea that it can be affordable. You don't necessarily need supply chain transport for it either. So we are going to continue to further develop SalivaDirect and saliva as it is. We do want to still reduce those diagnostic times and costs. So we have been exploring pooling strategies. We do want to move away from, or we realize that PCR is not, we're going to see we need to have a mix of testing options and a mix of avenues to go down. So, we're going to explore different technologies such as LAMP, and to sort of lead to the validation of point of care or in-home diagnostic platforms. Coming up to the flu season, we're also keenly aware of testing for co-infection. So having that dualplex PCR, we've got at least 3 other channels in there. So we do hope to be able to include the detection of at least flu A, flu B, and maybe RSV. And we are also interested in further exploring antibody detection in saliva, so that if you can, use a single sample type to test both for active infection through the RNA, or you could use it to investigate if there's been any past exposure. That would also be great. I wanted to just also share some of how things have been over the last few months, and sort of the lessons that we've learned. And that's one of the things, especially in the research lab, we just became keenly aware of the how to balance our needs as compared to the diagnostic needs. So really,
sorry to interrupt, you have 2 minutes.
Yeah, sure. This is, I think, the last slide. So being just very aware of our research needs and not detracting from the diagnostic needs. So really sort of developing our methods that weren't using things that were needed in clinical diagnostics. We also became very aware of our working schedules, which I think all of us are, especially very early on. We were very mindful to really schedule. I mean, we were in the lab throughout essentially all of this, but how we were sort of positioning ourselves, and also having, if there were key people for each step, sort of really staggering their roles so that you'd have minimal overlap, because we were certain that at some stage that we'd have an outbreak in lab or something from the community, not from our own practices, but we just wanted to make sure that some of the team was unfortunately taken down, which I'm really pleased to announce no one got sick, but just to have individuals trained on each aspect and sharing that knowledge and updating as you sort of do a handover of what's been going on. And it's also been so incredibly inspiring to see all of the national, international support and collaborations that have been going on, even just starting at Yale originally on the Impact Biorepository, and, working with people who I'd never even met before. And then suddenly, the number of calls we still have every single day of different groups, different labs, different even industry and commercial partners, just everyone sort of getting together to share ideas and experiences, and to really just help propel our knowledge further. This is not a time for us to sort of be very quiet about the progress that we're making, that, if we can already help others out, or say, hey, we've already done this, why don't you go from here? This is going to help us get answers faster than sort of each of us doing it in parallel, just very quietly. So it's been really, really inspiring to see everyone, or just so many people willing to do that. So I will now just wrap up with just an acknowledgement slide, and this is, you know, so many people who've been involved with the Impact Biorepository and also involved with the development of SalivaDirect, and yeah, it's just, you know, this is just mostly just from the Yale parts as well. So this doesn't even capture everyone that we have been working with, but I just really wanted to highlight just what a phenomenal sort of team effort this has been, and just how much everyone has just really thrown into this all. So yeah, I'm happy to take any questions.
Thank you for that wonderful seminar, Anne. I see that Saul Silverstein and Philip Protter have questions. If I could cajole them into raising their hands and asking them live, that would be great. And while I'm waiting for that, I'm gonna ask you a couple of, I think, relatively simple questions. What does it take for a lab to become a designated user of your protocol?
Yeah, so there's some, so first of all, they have to be a high complexity CLIA lab. And then so we have our own online form where they can request that. And then so far, you know, ideally they need to have the PCR instrumentation that we have on the EUA, but also if they have other instrumentation and they're willing to work with us on a bridging assay, we have all the lysates that we can send out to them. They can get that data, send it to us. And they do just need to, there's like a clear certification step that they have to do where they test a certain number of positive and negative samples through SalivaDirect. And we've just asked them to share that with us as well, just to make sure that, again, like, just to make sure that their process is going well. And if there's any apparent issues, then we can help sort of troubleshoot them. But that's really all it is.
Perfect. And how do you keep the designated laboratories up to date on the method as it evolves?
So far, we're very much relying on a Google form, but no, we're going to have a much more robust system of this in place. It's just been, you know, the last few weeks since the EUA have been wild, and just didn't realize everything that would have to go up behind us. But we're getting systems in place. You know, there'll be email lists that will have, like, you know, can send these out to individuals. It'll also be on our website. Yeah.
Perfect. Saul, you can
I'd like to know if you've tried isothermal amplification on the saliva test, because one of the roadblocks to rapid testing and widely available testing is the cost of the PCR machine for the test. And if that could be eliminated, that would be great.
We very much agree. So that's actually something that we've been exploring. One of the first things that we found is that the isothermal amplification can be it's a colorimetric. It can be very sensitive to the acidity of different saliva samples. So with one of the assays, we found that about 20% of samples would color change immediately. But we do know that there are other buffers out there that can help overcome that. We do know that there's just the, I believe, sorry, this is still a very new area for me, but more the fluorescence-based ones rather than the colorimetric ones. So that could also be a way of getting around that. It's definitely a option that we're very excited about, and we're starting to sort of explore now and get back into.
I will ask a question from Annette Ronska, and that question is, can eating or drinking before the test influence the results, and should it be avoided?
So our protocol, we just advise, like many of the other saliva protocols, is not to eat or drink or smoke, chew gum, clean teeth for 30 minutes before collection, and at the very most, like, don't try and avoid water for 10 minutes before collection. And saying that, some of the saliva samples that we received from the MBA were pretty much of every color of the rainbow, and so we actually had very robust RP detection still in those. So out of 3,800 saliva samples from the MBA, we only had 2 samples that came back as RP invalid. We haven't had an opportunity to go back and see why, but, you know, 2 invalid samples out of 3,800 I think is already quite a good success rate. So we aren't seeing you know, we haven't seen much evidence for what might, other than, as I said, unfortunately, maybe more mucus and such can interfere with the test.
Okay, and we'll give Philip another try. Are you there, Philip?
Okay, so I tried. Can you hear me now?
Yes.
Great. So my question is have you tested this saliva test against other kinds of tests, like the nasal test, to see the comparative accuracy, and have you done a statistical analysis? I
mean, so for the FDA EUA submission, we only needed to have the we had the 33 paired samples, and of course we saw the 94% comparison there. And, you know, we've done we saw high concordance with the MBA as well. But then again, they did such a great job at getting positives down that there were so few positives for us. So we have a very, very high agreement on our negative predictive value. And then we had high concordance with that. So I think we haven't done too many head-to-heads, but the fact that we developed this quite robustly in comparison to our samples that we were testing through RNA extraction methods. I mean, in the end, it's more about what's the viral load you're able to detect. And, you know, if that viral load is there, that low limit of detection, you know, it's more about that rather than, you know, I feel that if someone's like, you know, strongly positive or has low virus, like in their nasopharynx or in their nasal swab, you're going to get it in saliva as well. Again, you're going to see those discrepancies towards the lower levels of viral load where, you know, it can become more of a chance of picking enough viral material to detect it. And again, that's when, you know, if you have that frequent repeat testing, or at least, you know, at least twice a week, that's going to help you better catch, you know, maybe if you miss someone of low viral load one day, you'll catch them again as they start to increase in viral load in the coming days.
Okay. And the last question is coming from Bill Gruskin, I think the name is pronounced. It's related to the answer you just gave, and that is the virus initially detectable in saliva as soon as it's detectable in the nasal pharynx?
So that's an interesting one, because our strategy for testing the NBA samples is that if they flagged us with a positive nasal test, we actually went back and studied samples before and after in case there was anything else. And actually, both their anterior nares and oral swabs and our saliva samples were very aligned on detecting individuals at the same time. The only other thing that I found quite interesting was using RNA extraction, however, was when we were testing our asymptomatic healthcare workers through the impact biorepository. In a number of instances, we detected the asymptomatic healthcare workers first in their saliva samples, which could also, you can imagine, cause a bit of a stir at first, and you know how reliable this was. They were all confirmed by CLIA certified nasopharyngeal swabs afterwards. But it was interesting that we did find them first in their saliva samples as compared to the nasopharyngeal swab.